Welcome folks. In this video we are going to look at biology paper 2 for the 2011. Now we are going to focus on section A of this paper. We have figure 1.1 shows a plant cell which has been put in a concentrated salt solution. Now for them to use this language of in a concentrated, there has to be a comparison. So let's say we draw a diagram. We consider this beaker is where the concentration is high, so we have a high concentration. Then the question is what happens to the cell after being placed in a concentration which is higher than that of a cell. So that is the concept that we are looking at here. Now whenever we look at this one, we are going to look at the movement of water molecules from the cell into the solution, which is by osmosis process. Now let's maybe assign some numbers to these two solutions. So the solution that we have this one. Let's say this is a 20% salt solution. Then remember that a cell has also a concentration, has also a solution. And for example, we can say a cell has about 10% salt solution, which tells us that there is more, we have a lot of water molecules in a cell compared to the water molecules in a concentration, which is the solution. So what happens in our case is that the water will move from the cell into a solution. Then what happens if that occurs? So if that is the case, then we've seen the result of our cell. Now you need to, whenever you have a plant cell and an animal cell, please make sure that you differentiate this, because here we have the outermost part, which is the cell. Now for a plant cell, even if it loses water, the shape does not change, because the cellulose, which is the sugar solution here, is considered to be a rigid solution, so it remains in its original position. Then let's dive into the questions. Identify the parts about A and B. So A is a membrane that encloses the cytoplasm. So we have this cytoplasm here, and we know that whenever we are dealing with a fluid, we need to have a membrane that encloses that. So this membrane, its original position is here. Then because of the loss of water, we are looking at this membrane detach itself from the cellulose. So this membrane is the cell membrane. So the reason it has detached from its original position is because of the loss of water. Then for B, we have chloroplast. Then we move on to question B1. So what happened for the cell to reach this state? So you can say it has lost water through osmosis. Then set the term given to the cell in this condition. So we have plasmolysis. Then how can this condition be reversed? So by placing the cell in distilled water. So whenever we put it in distilled water, the cell will start now gaining water. Then give one reason why the structure labeled C remained in its natural state. For structure labeled C, we are looking at this one. So even after the losing of water, this structure did not change. So it's because of the presence of cellulose. So because of the presence of cellulose, which is rigid. Then we move on to question D. Give two differences between a plant cell and an animal cell. So the first one, difference number one, plant cell has cell wall, whereas animal cell does not. Then plant cell has chloroplast, while animal cell does not. Then there are so many differences we can also look at in terms of the shape and also the presence of the cellulose. Then we move on to question 2. Figure 2.1 shows an experiment to investigate the movement of curd solution in a plant. So we'll have the start, then finish. The arrow is showing us what happened after the movement of this solution. Then question A, name the process by which, Roman number 1, water in the solution was absorbed by the plant. So we have osmosis. Anything to do with movement of water is osmosis. Then the colored solute in the solution was absorbed by the plant. This is active transport. Active transport. Then we can also look at this one as also diffusion, since we are dealing with the particles. Why was oil put on top of the colored solution? So the investigation was that we only want the water molecules to pass through the plants to the leaves. So the reason why oil was put on top was to prevent evaporation of water. So to prevent evaporation of water. Then we move on to question C. Figure 2.2 shows the cross-section of the stem obtained from figure 2.1 at the end of the experiment. Then using letter X, label the part on figure which was stained by red dye. So red dye is considered as a solution, same as which vessel transports the solution. So it is this vessel. So we'll say XC is this one. Name, identify the parts you have labeled X on figure 2.2. So this is a xylem. So remember, xylem transports water and solutions. Name two substances transported by letter X and give one function for each. So the first one, water. Then a function for photosynthesis. Then also turgidity and firmness of the plant. Then the second one, we can use nitrate salts. So nitrate salts contains the nitrogen. So we'll say for synthesis of proteins. Then we move on to question 3. Figure 3.1 shows part of the respiratory system of an insect. Identify the parts labeled D. So D is where the entry of oxygen and the exit of carbon dioxide occurs through. So we'll have a spiracle. Then part E, we have muscle fiber, which is same as the tissue. Okay, we move on to explain how oxygen from the atmosphere is able to reach the tissue labeled E. So here you need to know that at point D, there is a high concentration of oxygen. Then here there is low concentration of oxygen, and the process of diffusion now must come in. And what is the use of this? So the use of this is that the oxygen, which is in a gaseous state, must be diffused. So it has to move from the point where it is in low concentration to a point, from the high concentration to the point where it is in a low concentration. So say oxygen will move from point D where it is in a high concentration to point E where it is in a lower concentration. Through the process of diffusion. Through the process of diffusion and through the tracheos. So this has to pass through the tracheos, remember that. So have the trachea, then tracheos. Explain how the energy released from the respiration in an insect is used. So we know that the insect does a certain work, and for the work to be done we need to have energy. So say energy is used for flying. So for flying we'll say which is locomotion, then we'll say for reproduction, and for metabolic reactions. So the use of energy that we are giving here is same applies to almost all the living organisms. We know that every living organism requires energy for that organism to do its work. Then set two ways in which the respiratory system shown differs from that of a fish. So we'll say respiratory of an insect, so of an insect obtains oxygen from air, whereas that of fish obtains oxygen from water. So for an insect, it is created in such a way that it can only get oxygen through air. That's why if you get an insect, then you, let's say, put it in water, it will not survive because the respiratory system is not meant for that. Then that it is also true for the fish. If you take the fish out of water, it will also not survive. Then the second one will say respiratory of an insect does not require circulatory. Wow. That of fish requires circulatory system. So what this simply means is that for respiration in insect, there is nothing like the oxygen must be transported by blood. For this one, after the oxygen has diffused into this insect, it will pass through the spiracle, then from the spiracle into the trachea, from the trachea into the tracheal, from the tracheal then into the tissues, which is the cells. So we can say that there is no transportation. So what we're saying is, for this one, we have the first one, spiracles, which is the entry, the passage. Then we have the trachea. From the trachea, we have the tracheal. After the tracheal, we have the tissue. But for fish, we are going to have, let's say the first one, the entry is the mouth, which is through water. Then we are going to have the gills. From the gills, then we are going to have the blood, which will transport the oxygen to the cells. We move on to question 4. Figure 4.1 shows the urinary system and its blood supply. So on figure 4.1, label structures F, G, and H. So F is just the blood vessel that is collecting blood from the parts of the body to the heart. So this is a vena cava. Then for G is a passage for urine from the kidney to the bladder for temporal storage. So this is ureter. For H, this one is just the passage for urine from the bladder to the external environment. So this is urethra. We move on to B. State 2 processes that are involved in urine formation. So the first one, we have ultra/pressure filtration. Then the second one is selective reabsorption. Okay, so we move on to question C. Table 4.1 shows the relative quantity of several substances in the blood in the renal artery and renal vein. Remember, renal artery brings blood, we say, toward the kidney. So this one is toward the kidney, then this one away from the kidney. So it's after the excretion is done. Then we can see that the quantities of these substances after the excretion was done, there is a reduction. For glucose 10.0 to 9.5, oxygen 100.0 to 40.0, sodium salts 32.0 to 27.0, urea 6.0 to 2.5, water 180.0 to 175.0. Explain what happens in the kidney to bring about the changes in the relative quantities of the first one, glucose. So the question is, why is it that the blood flowing in the renal vein has a lower concentration or has a lower level of glucose compared to that that found in the renal artery? So because as the blood is coming, it carries a lot of glucose. Then the glucose, a certain amount of glucose must be used by the cells that forms the kidney. Why? For that to do the work of excretion, that or the cells must have energy. So we'll say glucose is used by the cells of the kidney for energy purpose. So the key point is the kidney needs energy for that kidney to do its work. Then oxygen is used also, the same is used by the cells of the kidney during respiration. Okay, so we can also summarize for these 2 points as the same way glucose and oxygen is required by any other organ or part of the body, so is the kidney. So the kidney needs glucose for it to do the work, the kidney needs oxygen for that to do the work. So that's the reason why there is a lower concentration or level. Now for sodium salts is different. Sodium salts which is in excess is considered as a waste product. So say sodium salts in excess is considered as the waste products and must be removed by the kidney. Then for urea, there is nothing like excess. Urea is always considered as a waste product. So we'll say urea is a waste product which should be removed by the kidney. So that is, so for sodium salts and urea, these are removed because they are considered to be waste products. Then we move on to the last question, for question 5. In an experiment to demonstrate the inheritance of coat color in cattle, a roan bull was repeatedly crossed with a roan cow and produced roan, white, and road calves. Now, a situation where we have roan, this is ecodominance. Ecodominance in this case is where the two colors, or maybe two or more colors, are expressed at the same time. So red is seen, white is also seen. Then question A, which coat color is as a result of codominance? So which coat color is as a result of codominance? So this codominance brought about roan, which is a mixture of colors. So roan in our case will say white and red. Determine the genotypes of the three coats given that CR is a gene for red. Then, okay, so for the three coats, the first one, let's look at roan. So roan, we are saying it's a mixture, so we have CC. So C is first for, I'm sure, cattle. Then roan, we are going to have capital letter W, capital letter R. This is a roan. Then followed by red. For red, we need to have capital letters R. For white, the same, but we just have capital letters W. So the red and white must exist in the homozygous for a color to be expressed. Then question C, show using a genetic diagram the possible offspring which could be produced when a red bull was crossed with a roan cow. So we have red bull, roan cow. So we'll start with parental phenotype. So in our case, it's just the color. Here, the bull was roan. The bull, not roan, the bull is red. So let me just get rid of this. So it's red. Then for the cow, roan. So for a condition like this one, you just have to state a colors. Then a parental genotype. For red, it is CR, another CR. Roan, we have C white, CW, C red. Then we'll say parental gametes. We just have to split these. Then from there, we are going to have random fertilization. So this is our first one. So say genotype of F1, which is the generation of the offspring. Now, if for example, if the question was maybe another question to find a chance of producing Ron, so we have 1, 2, so it's 2 over 4 multiplied by 100%, giving us 50%. So for every random fertilization, there is a 50% of having a Ron, one of these then, and it is also true for red, there is also 50%. So that's it for this video. Thanks for watching.